Okay, then let us get started for today. So, welcome to the PMRF-NPTEL session on Introduction to Developmental Biology. This is week 12 and our final week, and today we are going to finish up our discussion on evolutionary biology and talk about different ways development can introduce variation that can be selected by evolution, as well as constraints that development might put on evolution itself. So I will briefly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur, and I'm taking these PMRF-NPTEL sessions. So as you would know, these sessions are designed so that we have an interaction with various students who are taking the course, so that we can solve various problems alongside them and better understand the concepts behind the problems as well as the course overall. So how we do these sessions is that I will show you a question from the previous year's quiz of the same course, and we will try to solve it together, discuss the solution, and then I will give you the answer and try to gain a lot of knowledge and conceptual understandings from all of this discussion. Okay, so let us get started for today. The first question is, which of the following can be explained by the reaction diffusion model? The options are formation of tooth cusp spots on the cheetah, the zebrafish stripes, tiger stripes, and eye color. This is a more than one option correct kind of a question, so you can choose more than one. I will give you a moment to figure this one out. Okay. So let us look at the reaction diffusion model. So the reaction diffusion model was proposed by Turing, and it tells us how very basic very basic biological reactions or chemical reactions can give rise to big asymmetries in a particularly uniform tissue. And this is also part of morphogenetic constraints. So how morphogens can set up a gradient in order to actually cause changes in a tissue. So how does it work? So say you have two different molecules in your tissue feed. One of them is P, which is a slow diffusing activator. What it means is that it activates itself, so it causes more of itself to be produced, as well as causes the production of another molecule called S. This S is a rapidly diffusing inhibitor. So as you can see, since P is slower, it has a higher peak where exactly it is produced, and then a very gentle slope as it goes further from the source. Whereas S, being a rapidly diffusing inhibitor, has a very shallow peak and very sharp, it has sharper slopes, or rather this one has sharper slopes and it has a shallow slopes because it is diffusing too rapidly around its own source, so it does not form a peak. So the reaction diffusion model actually tells us how exactly such a system can give rise to big asymmetries. So this is how it looks like at t equal to 0, say, where you have small peaks by random chances, where almost everything is mostly symmetrical. So now what will happen is that since P activates itself, it will cause more and more production of P at the source as well as on the sides as well. But since S is a rapidly diffusing inhibitor, what it will do is that it will inhibit P everywhere around its original source. Because the action of the inhibition is equal everywhere, it is just that P is just, there is a lot of P at the center. So after some time what will happen is that the peak of P will rise even further, because this is the only point at which P is being produced and autocatalyzed by itself, whereas everywhere else it is being inhibited by S. So you will have this huge peak coming up from a seemingly symmetrical tissue field. So this is what it looks like as a graph. Initially there is random changes, random peaks of P, but then because of the action of the inhibitor and the activator together, some of the peaks become isolated, and finally you have a structure like this where there is a huge asymmetry, where there are peaks which are present and everywhere else is completely flat. So this is how you achieve asymmetry. So this is how the reaction diffusion model tells us that from a symmetric tissue field you can generate asymmetric conditions so that tissue morphogenesis can take place in those regions. So where exactly do these things play a role? Actually anywhere where there is a formation of a seemingly random pattern, such as a lot of studies were devoted and applied the Turing model or the reaction diffusion model, and one of the major ones is the formation of teeth cusps. So this is our teeth. So it has two parts, as you would know. One of the parts is the bottom one, which goes into our jaw, and this is the one that you use for biting, and this top enamel part actually has cusps or crown like projection, as you can see right here. But what exactly determines where these cusps will form? So applying the diffusion reaction model, they figured out that it is actually that initially there is almost symmetrical tissue field, where little by little, due to the reaction interaction between FGF4 and sonic hedgehog, which are our slowly diffusing activator and rapidly diffusing inhibitor, we actually form these peaks where actually the cusps will form. And this is so these two things, so this one is our predicted shape and this one is our gene expression, and as you can see right here, the predicted shape and gene expression actually follow each other very well, both in mouse as well as vole, which is another rodent. So this just shows that this kind of a study, which is a computational study, can very well define biological problems, especially in this case. So if we go back to a question, which of the following can be explained by the reaction diffusion model? The answer is any kind of pattern which is seemingly randomly presented and needs to be made at the tissue level, such as formation of tooth cusps. The spots on the cheetah is also an example of the reaction diffusion model. Zebra stripes as well as tiger stripes, where there are random stripe patterns emerge from a originally symmetric tissue field. All of these can be explained by the reaction diffusion model. The eye color, on the other hand, is not such a pattern that can be explained by this model. Okay, so this was about the reaction diffusion model. We go on to the next question, which is choose the correct statements regarding mechanisms of evolutionary change. The options are: mutations responsible for differences between species majorly occur in protein coding sequences. Mutations in regulatory sequences are relatively dispensable. Mutations to create phenotypic variation occur in genes expressed during embryogenesis, and evolutionarily relevant phenotypic variations among species result from mutations in gene expressed in the adult stage. So this is a single correct kind of a question. I will give you a moment to figure this one out. And we are back. Okay, so let us look at it. So this concept is, so why are we talking about evolution in development? So as we know that evolution is essentially the survival of the fittest, one of the norms where nature actually selects the fittest fittest organisms that are part of a species, that have the variations that are needed to adapt to a newer environment. But the question is, how does evolution take place? So evolution takes place by introducing minute changes in the developmental program of different organisms. And how are these variations generated? So this is a very important question, because if development is complex and fine-tuned, as we have seen throughout our discussion in the course, how does it change without destroying the entire organism? So we have seen via various different examples how mutations or slight changes in sequences can give rise to huge debilitating final products where the organism cannot survive in nature by itself. So how exactly does evolution happen if development is so finely controlled? Because major changes or variations that need to be introduced at the level of initial early embryogenesis, because that is when the body plan is being set up. The answer is, there are different techniques that development and the different species or life as a whole utilizes to make sure that there are slight changes or variations that can help in evolution, but that does not change the overall plan of the body, so that you don't create fatal mutations or actually cause many different problems. So if variations arise from mutations in protein coding genes, there will be mutant protein that will be expressed in all places where the protein is normally expressed. This should be one of the key ideas on why development needs, like this is one of the key ideas which follows the question right here, that if say to generate a very slight variation, you change the protein that actually is responsible for that particular morphogenetic, like morphogenesis of that part of the body, but then wherever the protein is expressed, it will change everything else. So how exactly do you deal with that? How do you tweak the developmental program so that you only have variations, but your overall organism is safe from actually having a completely wrong body plan? And the answer is modularity and molecular parsimony. these are things that we have discussed in last week's class as well. Modularity is that the idea that there are different anatomical and DNA region modules, and these modules can independently act, and when you place the module somewhere, it will generate a particular functional part that can be used to introduce different variations. Such as examples of anatomical modules include morphogenetic fields, cell lineages, or insect parasegments, where if you change morphogenetic fields, such as say change the field which gives rise to the limb section, the limb regions, then you can have different types of limbs that are present in our different organisms. And then there are DNA region modules, which I've already seen, which are enhancers. So you can just plug and play enhancers, pick it up from somewhere, put it in somewhere else, and the evolution enhancer will work almost similarly, so that you can have tissue specific expression changes throughout development, which will generate variations which can be selected by evolution. So this is one of the ideas behind how development contributes towards evolution. The other one is molecular parsimony. So what does that mean? It means that development occurs by interaction of a very select few genetic pathways or players, and these ones are present throughout the body doing different functions. So it is the same toolkit, as they say, which you can use to build a bicycle or an airplane. And so what this does is that different like it is very easy to introduce changes in a particular body plan when you are using the same screwdriver for everything. So that is where molecular parsimony comes from. So for example, the toolkit pathways such as the BMPs, the FGFs, the serine threonine kinase, anything. So all of these are used everywhere in the body, and changes to one of them can cause various changes in the body plan. So that gives rise to variations, as well as other master regulator genes such as the Hogs genes or the homeotic genes. If you cause changes in them, they also give rise to huge changes in the body plan, and that is all because these genes are so powerful, as well as that they are so few of them that you can tweak them around. But the development has also figured out a way so that variations can be introduced without major problems. So such important genes such as for example, the homeotic genes actually have a number of paralogs. So HOX-A, which is present in vertebrates, actually there are multiple HOX-As. So there are HOX-A1, HOX-A2, HOX-A3, and there are similarly all the way to HOX-A13. So minute changes to individual ones can give rise to variations, but if there is a detrimental problem that comes up from these changes, then the paralogs can actually redundantly rescue such a phenotype, and thus your organism body plan remains the same. Thus, having a number of copies of the same genes help in maintaining molecular parsimony, as well as allowing development to contribute towards evolution. And what are the different mechanisms of evolutionary change? These are heterotopy, where you take a particular protein and make it cause it to be expressed somewhere else. Heterochrony, where you take a protein and cause it to be expressed at a different time in development, or for a longer or shorter period of time during development. Heterometry, where you change the amount. And heterotopy, which is actually changes in the amino acid sequence, and then you change the function of the protein, and it does something new entirely now. So these are the different techniques in which early development can contribute towards evolution. So choose the correct statement regarding mechanism of evolutionary change. The correct option is mutations to create phenotypic variation occur in genes expressed during embryogenesis, because later on in different organisms, then the genes being expressed do not cause changes in morphogenesis or changes in the body plan itself, which are required for adaptation. For example, say a particular bird will which needs to adapt to a new environment, from being a hunter or a bird of prey to a bird which eats seeds or cracks seeds, needs to have modification in its beak. this modification in its beak does not happen once the bird is now an adult bird. So it will not start changing its gene expression so that its beak converts from a sharper to a blunt and stronger beak. It will not happen like that. What will happen is that over generations, early embryonic changes will take place, which will cause some of the individuals of the species to have stronger and blunter beaks, which are more capable of crushing seeds, and these individuals will take forward these changes and will be selected by evolution, and that is how over generations you have adaptation. So essentially all the phenotypic variations that we see during in different species is during embryogenesis. Okay, so moving on, the next question is, which of the following mechanisms are true regarding heterotopy? So heterotopy, the change in location of gene expression. The options are: BMPs are responsible for interdigital apoptosis in bats, gremlin antagonizes BMPs in bat wing, dermally expressed FGF10 attracts Rib precursors, BMPs are expressed interdigitally in all vertebrates, and mice deficient in BMPs will retain interdigital webbing. So there are 5 options and this is more than one correct option. So I will again give you a minute to figure this one out. Okay, let us look at it. So as we said, heterotopy is uh, heterotopy is the expression of a protein at a different location than where it normally occurs. So the example we are going to deal with for this is the formation of the bat wing. So as you see, in vertebrates such as the mouse and in humans as well, we have 5 different fingers. We do not have anything in between. But during early development, as you can see, the limb bud, which will give rise to our entire limb as well as the digits, actually is kind of a spherical structure. But as development proceeds, this particular you can see digit like projections in this limb bud, and there something interesting happens. This webbing in between the fingers actually apoptosizes, so all the cells die off so that we have separate fingers. This happens in mice as well as in humans, and finally we have different fingers as the adult body. So that is why mice have different fingers, humans also have. They do not have anything in between the fingers joining them. But in case of bats, something interesting happens. So if we look at it in some detail, you will see the bat's wings, the bones in the bat's wings are actually the phalanges of the fingers. So as you can see, these are fingers which have grown a lot longer than in case of mice. And The interdigital mesenchyme which is present, which in case of mice apoptosizes and dies off, causing separation of the fingers, actually does not do so and develops instead into the leathery bat wing. And this can be observed in the developmental time scale as well. So initially bat also has a spherical kind of a limb bud which will give rise to digits. At a stage such as this, you have the digits being prominent, but as growth occurs, the interdigital mesenchyme does not die or does not apoptosize, and thus it remains and you have the bat wing. Something similar happens in case of ducks as well, where they have flippers because of the same reason, because the interdigital mesenchyme stays and develops into the flippers. Now this is a very prime example of heterotopy, because this dying of the interdigital mesenchyme in case of mice and humans as well occurs because BMPs are being expressed in this region, and that is why they die off. But in case of bats, the bats express another signaling molecule FGF8 in this interdigital mesenchyme as well. This causes this interdigital mesenchyme to not apoptosize and be able to develop into an entire bat wing. So heterotopy, expression of a molecule where it is not usually expressed, giving rise to variations and development of new features. FGF8 is now being expressed in these regions where it is normally not, and this gives rise to the bat wing. So heterotopy. Another example of heterotopy can be cited as the development of the turtle shell. So the turtle shell is actually the entire rib cage of the turtle, which instead of growing towards from the dorsal to the ventral end actually elongates and forms the entire shell. So what happens is that at an early stage, this is our rib, this is the turtle rib, and instead of curling around and covering the rib, the heart and other organs, there is instead an expression of FGF10 towards the distal end. This FGF10 actually causes the ribs, instead of growing in a curved fashion, to grow straight and then develop into this rib-like structure which supports the shell. And this ribs, when they grow, they actually also carry BMPs with them, and what BMP does is that it causes the skin on the back of the turtle to also form into bones, and that is how the entire shell is formed. So the BMPs from the ribs actually cause the entire back of the turtle to convert into bones, and that is why you have a bony shell on the back of the turtle. So again, this is an example of heterotopy in which FGF8 is expressed where it normally is not. The location is changed, which gives rise to a completely new phenotype. And again, this is an example of how development can affect evolution. So that was that. So which of the following mechanisms are true regarding heterotopy? BMPs are responsible for interdigital apoptosis in bats, but it is opposed by FGF8. Gremlin antagonizes BMPs in bat wing. Dermally expressed FGF10 attracts rib precursors, this we just saw. BMPs are expressed interdigitally in all vertebrates. That is also true. Okay, so all of this are examples of heterotopy, and we move on. The next question is, match the following expression patterns with their beaks. So here we are talking about Darwin finches. So if you have gone through that paragraph of the lecture, this might be easy, otherwise we'll look at it. So they have told us that in case of embryo A, we have small amount of BMP4, high amount of CAM, which is calmodulin. Embryo B, high amount of BMP4, low amount of calmodulin. And embryo C, moderate amount of BMP4 and low amount of calmodulin. And we have 1, 2, 3, 4, 5, 6 options on how will the beak develop in case of all these embryos. So for example, the option A says that the embryo A will have narrow and long beak, the embryo B will have wide and short, and the embryo C will have medium width and short beak. So I will give you a moment to figure this one out, and try and answer how exactly the expression of BMP4 and calmodulin might affect the beak length in case of these finches. So you have only one option correct here, A, B, C, D, E, F, and I will give you a moment to try and figure this out. চা দাও अंजा बोल दिया जल्दी जाओ এরা কথা বলছে Okay, so let us get back to it. Here we will talk about how about a particular type of bird which are known as Darwin finches, and how exactly the expression of BMPs can describe how exactly development occurred and evolution per se occurred in these particular finches. So these were discovered by, not discovered, but like researched by Darwin as he traveled to the Galapagos Islands, and he saw very related bird species which differed only in the size of their beaks and the kind of food they had. So some of the birds which were feeding on cactuses actually had longer beaks which could reach past the spikes of the cactus, whereas other ones which bit into harder fruits and seeds actually had blunter and stronger beaks. All of this can be explained by heterometry, which is the amount of a protein being expressed. So later on studies were done and people figured out that the expression of BMP4 actually determined how thick the beak or can be in case of these particular birds. So wherever there is a very high expression of BMP, the beaks are narrower, as you can see right here, and then as we have lower and lower expression of BMP, we have thicker beaks. There is a similar thing can be seen in case of calmodulin. More calmodulin being expressed, you have longer beaks. So as you can see right here, as the expression of calmodulin increases, you have longer and longer beaks. And taken together, people finally figured out that the length and width of the beak actually was determined by BMP and calmodulin expression, which was the developmental and molecular understanding behind how these different species evolved. So when you have low BMP, then you have low beak width. So low BMP you have low beak width, and high calmodulin you have an elongated beak, which is in case of the cactus finch. Then you have low to moderate BMP, you have a moderate beak width, and high calmodulin, which you again have elongated beak, but this time it is thicker. Moderate BMP and low calmodulin, you have a shorter beak, but it is still thick. And then you have a high BMP, which gives rise to a very thick beak, and low calmodulin, which gives rise to a short beak, so it is a strong and short jaw. So this is how, and all of these adaptations developed because the different species had different eating habits, such as the sharper and longer beaks belong to finches, which went into probing into cactus flowers and tried to avoid the different needles on the cactus and go inside the flower, whereas crushing seeds was done by bird species which had stronger beaks, which were blunt and thus stronger in order to break all of these seeds. Okay, after learning all of that, let us look at how exactly we can predict what is the type of beak a particular finch will have based on the expression of BMP in calmodulin. So the first thing is that BMP is low, which will tell us that the embryo A will have a very narrow beak, and calmodulin is high, so it will have a longer beak. So we are looking for narrow and long, and we only have one option which is correct, which is A, narrow and long. And the second one, BMP is high, which is a wider beak, whereas calmodulin is low, so wide and short. And finally in embryo C, it is medium BMP, which gives rise to medium width, which is part C, and calmodulin is low, so it will be short. So medium width and short, so all of them taken together, the option A is the correct one. Okay, next, if A is heterochrony, which is changes in time, and B is heterometry, which is changes in amount, which statements are correct? The options are sonic so options are that B is sonic hedgehog overexpression causing down regulation of PAC6, or B is sonic hedgehog overexpression mediated degeneration of the eyes, A is FGF8 mediated elongation of the middle digit of the dolphin flipper, or A is sonic hedgehog mediated production of digits in the lizard. Again, this is more than one option correct kind of a question, so I will give you a moment to try and figure this one out. Okay, let's look at it. So heterochrony, we are going to look at heterochrony for a bit now. So as we said, heterochrony is a shift in the relative timing of developmental processes. So change in relative time may be either in the development of a particular model or in gene expression. For example, we know that snakes have a very long anterior posterior axis, so they're long organisms where the entire spinal cord has vertebrae which forms ribs. But the question is, how exactly does the development of the other parts of the body occur in the same time as formation of so many ribs throughout the spinal cord? The reason is, in case of snake rib development, the segmentation reactions cycle nearly 4 times faster relative to tissue growth of the embryo than they do in relative vertebrates. So essentially, in case of snakes, the formation of the ribs occurs 4 times as fast as development of any other structure, so that development can keep pace with uh, the development of the other tissues can keep pace with development of the ribs along the spinal cord. Another example is hyperphalangy of digits 2 and digit 3 of dolphins. So dolphins, we know, have flippers that help them go around and swim, but these flippers are actually formed by extending the phalanges of the digits of digit 2 and digit 3 in dolphin. So what exactly, like how exactly does development of the digits keep pace with the development of other parts of the body, where we have like a huge phalange forming rather than the normal shorter one in case of other vertebrates? The answer is that this is again heterochrony taking place, where the growth of the phalanges occurs really fast as compared to other tissues in the body, which causes the growth of the phalanges to be keep pace with the development of the body. So again, there is change in relative timing of the development processes, which is heterochrony. This is another example of the same in case of heterochronic hypothesis for cetacean limb development. Cetaceans are dolphins and whale-like creatures, which are mammals which breathe air but live in the sea. And in their case, as I said, hyperphalangy of digit 2 and digit 3 occurs, in which you have a lot more phalanges than normal, and this development occurs really fast to keep pace with the other parts of the body, and is regulated by sonic hedgehog expression. Next, we look at heterometry, which is changes amount of expression of a particular gene. So for this one, we look at the formation of eyes. So the formation of eyes, we have looked at it briefly earlier in previous lectures, where we saw that PAC6 is the particular master regulator of formation of eyes. So wherever PAC6 is expressed, eyes will try to form there. And initially what happens is that there is an expression of sonic hedgehog, which divides, which goes along this axis. So it is somewhere here where sonic hedgehog is expressed, which causes the eyes to there for so it what it does is that it inhibits PAC6 expression along the middle of the face, so that when PAC6 is expressed, you have two eyes, because you have a line of sonic hedgehog going in the middle. So two eyes instead of one big eye. But in case of a particular kind of fish, which is Mexican tetras, there are two variants of the fish, where one of them stay on the surface of the ocean, and there they have their normal eyes and expression of sonic hedgehog and PAC6. But then there is another variant of this particular species which live in deep dark caves. In these kind of fish, they have actually lost their eyes because of an increased expression of sonic hedgehog, which abolishes any expression of PAC6, and thus you do not have eyes in these kind of fishes. So you can see right here, PAC6 expression occurs in these two regions, which gives rise to our eyes, and these two regions are missing in case of the cave dwelling fishes. And the reason for this kind of evolution is that the cave dwelling fishes actually develop other senses to figure out where they are in the dark cave environment, which need a lot more resources, and they gave up their eyes in order to push the resources towards that end and form the other structures. And all of this is contributed by heterometry, which is change in the amount of sonic hedgehog, which causes abolishment of the eyes and allows more and more input to go towards development of other senses. Okay, so going back, if A is heterochrony and B is heterometry, which statements are correct? So B could be overexpression causing downregulation of PAC6, which is heterometry as we saw. B can also be a sonic hedgehog overexpression mediated degeneration of the eyes. A could be FGF8 mediated elongation of the middle digit of the dolphin flipper, which we saw as well. and A could also be sonic hedgehog mediated production of digits in a limb. So in lizards. So in case of lizards, the number and the length of the digits is determined for how long sonic hedgehog is expressed in the limb, and so A could also so this is also an example of heterochrony. Okay, moving on. The following gene is divided into 6 modules A to F. If a mutation occurs simultaneously in 2 modules, which pairs are both likely to cause a heterotopy? So you have division of the particular gene in A, B, C, D, E, F. So essentially, which of these will cause heterotopy or a change in function? And here you have to make sure that you look at the word both. So you need both the options to cause heterotopy in order for that option to be chosen. So you have different option A, F, A, B and different pairs. So I will give you a moment to try and figure this one out. Which two will always cause heterotopy? And this is more than one option correct kind of a question. So you can choose more than one options here. Okay, I will give you a moment to figure this one out. Okay, let us look at it. So what is heterotopy? Heterotopy, as I said, is mutation that actually alter the amino acid sequences of the protein. So these need to be specifically in the exon part of the mRNA. Any changes in the exon can actually give rise to heterotopy. So let us. So here's an example of heterotopy. So in case of insects, we know that insects actually have a lot more legs than vertebrates that we see around us, which are mostly tetrapods, 4 legs. But in case of Drosophila, say they have 6. Centipedes and millipedes have much more. And why does this happen so? So the reason being, in when the ultrawide thorax sequence domain was sequenced and analyzed for different insects, they figured out that there is a stretch of polyalenine in these particular insects that are present, and this they attributed towards causing this increase in number of legs, because there were changes in the UB, the ultrabithorax peptide, which causes it to form more and more legs and more segments in case of these insects. So only in insects, ultrabithorax protein inhibits the distalless gene. This is because of the poly A tail that is added in these particular insects, and distalless causes shortening of the number of limbs. So since ultrabithorax starts inhibiting it, you have so many limbs in insects. So now let us look at it, which of these are going to cause a heterotopy. As I said, this will if a heterotopy has to occur, then there need to be changes in the exons, because they are the only ones that are going to be translated into proteins. So the options here are A and F. A being in the promoter region, no exon are present, so this we can just not take into account. A and B, again A is present, so again, so both of them will not cause a heterotopy. Next we have B and E. B, as you can see, spans a exon. কি হই দিয়ে দাও না, আমাকে কেন মাথা খাচ্ছ এখানে বসে? B and E, you see B spans an exon and E also does it, so it can cause a heterotopy because there can be changes here which add translation initiation codon early on. Changes in protein sequences will be there. Similarly, changes in the stop codon will cause extension of the protein sequence, and that also changes the protein sequence. So B and E qualifies for heterotopy. C and E also does it. C just spans the exon, and E has the same thing as I said. C and F. So C spans the exon, so it could qualify, but F is present after the termination codon, and heterotopy requires changes in the protein sequence, so F can never give rise to changes in protein sequence, so it is removed from this. And D and F also have F, so even if D does a heterotopy, both of them cannot do heterotopies, so only these two options are the correct one. Okay, moving on. Select and the correctly matched pairs in these two options. On the left we have three different constraints of development on evolution, which is physical constraint, morphogenetic constraint, and phyletic constraint. On the right we have different options which can go along these. Here we have to choose which of these actually match up with which of these. So a simple match the following, and here are the different options for them, and this is more than one option correct kind of a question, so I will give you a moment to figure this out. Okay, let's look at it. So there are different constraints, developmental constraints on evolution. So as we said, variations that can give rise to evolution can only be small tweaks to the entire structure, and cannot be a big change that occurs and can destroy development itself. So these are because there are different constraints put on evolution by development. And there are three major types: physical constraints, morphogenetic constraints, and phyletic constraints. So physical constraints include just laws of physics which govern normal development, such as law of diffusion. Hydraulics and physical supports are immutable. Blood cannot circulate in a rotating organ. 6 foot tall mosquitoes and 25-inch long feet long leeches cannot exist, because all of them defy basic laws of physics, and thus cannot be possible for evolution to just for organisms to adapt and evolve into such structures. Next, morphogenetic constraints. We have touched upon this previously. So although there have been many modifications of the vertebrate limb over the 300 million years, some modifications such as the middle digit shorter than its surrounding digits are never seen. So all of our we see that in our hands, the middle digit is always longer than the other digits, which shows that there must be some constraint that causes this to occur the same across species. And this is the morphogenetic constraint that we talked about using the reaction diffusion model, where we saw that in only particular points we can in a symmetric field or in a symmetric tissue field, we will have particular points where asymmetry will occur and morphogenesis will take place. And this morphogenesis actually is the reason for why our fingers appear where they appear, and also because a reason why our middle finger is longer than the other ones. So these morphogenetic constraints also play a role in evolution. And finally, we have phyletic constraints, which are historical restrictions based on genetics of development. So presence of notochord, which is still present in vertebrates but actually do not have any adult functions, are still there in vertebrates. Lack of variation in limbs, constraints on alternate body plans for pleiotropic nature of insect segment plurality genes, involvement of Hox genes that specify cervical vertebrate stem cell for proliferation. These are constraints uh, imposed by inductive effects of the phylotypic stage. So what is this? So as we know that organisms of a particular phyla all have a very common phyletic stage. So in case of in the vertebrata or the vertebrates, this particular stage is the late neural stage known as the pharyngula, and this stage looks very similar across the entire phyla. So essentially, as we have seen before, that the early embryo of different species actually look very similar, and species specific changes occur later during development. And this is a reason for that, because all of the development proceeds very similarly up to the phyletic stage, and later on different structures develop requiring different depending on what species the pharyngula belongs to, or what species the organism belongs to. But this also makes sure there is a constraint, because you can't have different structures which were not present in the pharyngula or the phyletic stage. Structures which are not compatible with the phyletic stage appear in that particular phyla, because the body plan will start from this stage only. So you can't just introduce something new which cannot develop from this. So this is a phyletic constraint. So let us now look at the different options. The correct options are the first one and the second one. The first one is physical constraint goes to 1, that the idea of a homunculus is impossible. So homunculus is the idea that our bodies are predefined and pre-built at a very early stage, and we just grow instead of developing into new organisms, like developing and forming new structures. We just grow, and that is not possible because of various physical laws, which is the physical constraint. B is 4, which is morphogenetic constraint, unidirectional development of humerus and radius and ulna and hand. This is because the direction in which the morphogenetic gradient is set up, and thus it is a morphogenetic constraint that our humerus cannot grow the other way around. And C is part 3. The finite constraint is phalangular stage. We have already seen this. The other option which is correct is A is 2. Physical constraint, secondary growth in the tree, which can occur only, so there is secondary growth at only a particular point, which is also follows the laws of physics. B is 7, which is a lower cell number forms a limb with fewer digits. This is obviously a morphogenetic constraint as well as a physical constraint. And C is 5, which is finity constraints the compartmentalization of instructions into organ forming systems at later stages of development, which is a finity constraint. Okay, moving on. Which of the following explains the frequent co-occurrence of an extracervical rib and embryonic cancers? The options are: genes involved in body patterning also play a role in regulation of stem cell proliferation, phyletic constraints playing a role in development and evolution, extracervical rims directly cause embryonic tumor, embryonic cancers directly cause the growth of an extracervical limb. This is a more than one option correct kind of a question, so I will give you a moment to try and figure this out. Okay, so the question is, why are extracervical limbs connected to childhood cancers? So is uh, this is again another example of how heterotopy has occurred. And so heterotopy is the changes in the function of a particular gene. So in case of particular one of the Hox genes, one of the genes required for vertebrate development, is also associated with stem cell proliferation. So people observe that when they are looking at early embryonic cancers or early childhood cancers, they saw that many of the patients actually also had an extra pair or some more cervical ribs, and these occurred majorly in cancer patients but did not occur in the control or the general group. So whoever seemingly had, so they seem to be developing a correlation between extra number of cervical vertebrae and embryonic or childhood cancers. So they tried to figure it out, and they finally figured that there was a particular Hox gene which was related to stem cell proliferation as well as growth and development of cervical vertebrae. We have seen that how Hox genes play a role in development of vertebrae as well as patterning them early on during the previous week's lectures. And heterotopy actually played a role here, where the Hox or heterotopy played a role here or rather phyletic constraints played a role here, where the gene responsible for stem cell proliferation, when it is mutated, it also causes, because it is the same gene, causes extra number of vertebrae to form, and also causes cancers to appear in early childhood. So essentially, extra if extra vertebrae occur, are detected, they are surefire indication of there being any changes in the gene behind it, and that gene also helps support stem cells proliferation, which can give rise to cancers, which we see right here. So the correct options are: genes involved in body patterning also play a role in regulation of stem cell proliferation, and phyletic constraints playing a role in development and evolution. Next, select the correct statements regarding epigenetic inheritance systems. The options are: it allows environmentally induced changes to be inherited, epialleles are variants of DNA sequences, epiallelic modifications occurs in the germline, hypermethylation of a gene will have the same effect as loss of function gene mutation. So I will give a moment to try and figure this one out. Okay, let us have a look at it. So this, we have seen how changes in the protein coding sequences, as well as the genomics, genetic sequences, or the gene sequences can play a role in, or the changes in the DNA sequences can play a role in evolution. But there is also this idea of epigenetic inheritance system. So changes in something not directly being the DNA sequence, but actually causes changes in development, variations in development, which gives rise to evolution. So there is this idea of developmental plasticity, which says that development of alternate phenotypes or polyphenism can occur with the same genotype or the same DNA sequence, but you will have different genotypes, phenotypes that occur, and the specific phenotype being dependent on the environment. Since such changes in development do not occur due to changes in the DNA sequence itself, it is called epigenetic variation. So developmental plasticity allows for epigenetic variations, which is stable in some instances between generations. So these epigenetic changes can be inherited, and these are known as epigenetic inheritance systems. So for example, there are majorly three types of epigenetic inheritance systems. The first one is epialleles, which are they not, so in this case variations in DNA structures don't occur, but chromatin structure does occur, one of them being DNA methylation patterns. So in case of toad flax, so we have one flower phenotype where there is relative unmethylation in the particular flowering gene, but in another the flowering gene is highly methylated, and that is why you have a different flower phenotype. Again, this is not a change in the DNA sequence, but a change in the chromatin structure. So this is a epiallele or epigenetic type of inheritance. The other being symbiotic variation. So this is when two organisms live together and help and support each other, and changes in the genotype of one can be inherited and cause phenotypes in the other. One of the examples could be the microbiota in case of humans. So we have a huge intestinal microbiota which helps us digest as well as contributes towards various needs of our body, and any changes in that can actually cause changes in the phenotype of the humans, and these changes occur by changes in relative abundance of a particular symbiotic organism, introduction of a new symbiote, changes in the microbial genome, and this changes in the microbial genome can occur really fast because microbes have a shorter DNA and much less amount of genes, and chances of random mutation and crossovers is much higher. And this can occur within one generation of a particular organism. So for a particular human, the throughout its lifetime, the microbiota can change very drastically, having newer mutations in some time, and this occurs within one generation, which normally would have, and changes thus of the phenotype also occur in one generation, which normally would have taken multiple generations to occur. And horizontal gene transfer is possible between microbes, so such changes can be propagated, which are beneficial to both the host and the symbiote. And this is possible because microbes can transfer genes horizontally. So this is changes in symbiote. And for example, these are two mice which are fed exactly the same amount of food. So what is special with the mice at the right is that this mice had a leptin mutation, which caused the gut bacteria to become super efficient at absorbing nutrients from food. So with equal amount of food being given to the two mice, this one can gain a lot of weight because the food it consumed was completely like much more nutrients were absorbed from that food, causing it to gain a lot more fat as compared to the control mice. And when the gut bacteria from this particular leptin mutant mice were taken and introduced to a normal control mice, this can also cause rise in fat accumulation in the control mice, which shows that this kind of symbiotic changes can be inherited between different generations as well as individuals, where there is not the initial genetic trigger is lost. So this control mice, after the bacteria is transferred to this, can also gain fat, but it does not have the leptin mutation that this mice actually had, which triggered the bacteria by microbiota to change. And thus this is epigenetic kind of variation, where this mice inherited a quality with no changes to the genotype. And finally, we have genetic assimilation. This is the process by which a phenotypic character initially produced only in response to some environmental influence becomes, through a process of selection, taken over by genotype. So that is formed even in the absence of the environmental influence that had first been necessary. In short, phenotype precedes the genotype. So what all of this means is that, as we saw, polyphenism is the same genotype, so same DNA sequence can form different phenotypes. And for example, if you have ever seen, very basic example is that there are particular dogs that are present, like you have seen dogs all around the world, but stray dogs which live in cities like ours, which are nearer to the sea level, none of them have fur. They look very lean and they go around their happy ways. But if you go further up the mountains where it is way colder, you will see dogs will always have this thick fur to actually combat this colder weather. So this is the same species. So where does this change appear from? So what has happened is that these organisms always had the genes to form longer coats or smaller coats, but depending on the environment, now a particular trait is being more and more suppressed, and another trait is being uh, activated, and which causes the dogs living up in the mountains to have thicker coat. So now once these particular dogs or any other organism has this trait, this is chosen to be the fittest, and this adaptation is carried on through generations. So the next generation will always have the thicker coat, and there will be no need for new mutations to occur to form the bigger coat. Something similar happens in case of an experiment done on Drosophila, in which uh, the scientists looked at Drosophila, which were embryos which were kept in ether, and some of them developed a mutation which causes four wings, which is the bithorax mutation in the alter bithorax complex. So they now have four wings. So these two normal wings are cut in this image. So actually the fly had 1, 2, 3 and 4 wings. What they did is that they took flies with four wings and mated them exclusively with flies with four wings as well. And in the experiment, the flies with two wings were mated with flies with two wings. And when selected for this particular trait, they saw that finally, that in the after multiple generation, the four-winged phenotype was retained in the Drosophila which were mated for that or selected for that phenotype. So they first observed the phenotypic change and selected for it, and they saw that individuals expressing that trait increased as they kept on selecting for it. And similar thing happened in the other way around. The Drosophila in which they only selected for two-wing trait actually showed two-winged phenotype as they went further and further across generations. We showed that once a particular phenotype is evident, you can select for it, and that particular selection will be genetically assimilated in the population. So this essentially you select for that trait, and that is the trait the most of the population will show later on. So this is known as genetic assimilation. So select the correct statements regarding epigenetic inheritance systems. The answer is epiallelic modifications occur in the germline, which is obviously the case because otherwise it cannot be inherited across generations. And hypermethylation of a gene has the same effect as loss of function of gene mutation. This we have seen seen in classes way earlier, where hypermethylation gives rise to loss of function. And this is also epigenetic inheritance kind of a thing because no changes in the actual DNA sequences occur. Okay, next. Which of the following statements are correct in the context of genetic assimilation? The selection of dark-colored moths, as described in industrial melanism, is prime example of genetic assimilation. Genetic assimilation can cause intraspecific differences in phenotype. It enables adaptation. It requires initial mutagenesis to develop the phenotype, and it is enabled by the developmental plasticity of structures rather than induction of new structures. So let us look at it. So we have seen genetic assimilation. It is the process by which a particular change in phenotype is induced by the environment, and that the organism which has that change is now selected as the fittest, and that organism passes on the trait to the next generation, and now the next generation slowly has changes in its genotype which causes that particular trait to be the dominant one compared to everything else. We saw that in case of the bithorax mutant, when only four-winged flies were selected for that four-winged phenotype, the number of individuals in the population that showed the four-winged phenotype increased as we went past generations. So this is genetic assimilation. Similar thing has been observed in case of in nature as well, in where a particular species of butterfly have changes in the pattern on the wings depending on the temperature of its surrounding. So a similar thing, similar example as we discussed before. So depending where the butterfly is from, the pattern on its wing changes, and this is again an example of genetic assimilation where a phenotypic environmental trigger, which is the temperature, induced a change, and that trait, change in genotype was assimilated as we went along generations, so that a particular butterfly from a particular region will have that pattern only. So the phenotype of a species that is characteristic of a geographic area is called ecotype. And the other thing is that environmentally induced phenotypes or genetic assimilation are really important aspects of evolution, because the phenotype is not random. The phenotype was always hidden but suppressed because the environmental trigger was not present, but since it is always present, it shows that it is not harmful to development, so it will not cause fatal changes in early development. And the phenotype already exists in large portion of the population. It is just suppressed because of the other dominant phenotype. and whenever there is the environmental trigger, major part of the population can adapt really fast in the span of a single generation. So you don't have to wait for multiple generations for any adaptation to occur. Okay, so all the options were correct in this particular question. The selection of dark colored moths as described by industrial melanism is a prime example of genetic assimilation. Genetic assimilation can cause intraspecific differences in phenotype. It enables adaptation. It requires an initial mutagenesis to develop the phenotype. It is enabled by the developmental plasticity of structures rather than induction of new structures. Okay, so that is all I had for you today, and this is the last class of this particular course. I thank you for attending this particular course throughout, and if you have any questions, then you can always put them in the YouTube channel comments, or go to the NPTEL forum and press them in, and we can have a discussion after that. I wish you all the best for the NPTEL exam as well as various future interviews. Thank you.